Well, we've made it to the last session of our meeting. In this session, we will hear updates from 2 additional Cohort Consortium groups, project groups. I'm Jeannette Schenk from the Fred Hutch Cancer Research Center, and I will be co-moderating this session with Dr. Nonye Harvey from the NCI. We're going to go through both of the talks first and save our last 10 minutes or so for questions at the end. Our first group update will be given by Dr. Hilary Robbins. Dr. Robbins is an epidemiologist at the International Agency for Research on Cancer. Her research focuses on the development and application of risk-based approaches to cancer screening, in particular for lung and HPV-related cancers. Today, she will be providing an update on the Lung Cancer Cohort Consortium project and their efforts to develop a biomarker for early lung cancer detection. With that, I'll give it over to you, Dr. Robbins.
All right, can you hear me okay? So I will be presenting an update on the work of the Lung Cancer Cohort Consortium, in which we are developing a protein biomarker panel for early lung cancer detection. I'm collating this project together with Matthias Johansson, and I also want to give a lot of credit to the other members of our team who are shown here, who made really important contributions to this work. Next, please. The motivation for this work is really rooted in the context of lung cancer screening. There are now 2 large randomized trials showing that low-dose CT screening for lung cancer can reduce lung cancer mortality among people at high risk. So these are current and former smokers with a heavy smoking history, usually aged over 55. The first trial was the US National Lung Screening Trial in 2011, and then earlier this year, the Dutch-Belgian Nelson trial was published as well. Next, please. And lung cancer screening has really become sort of a poster child for the idea that we can use information about individual lung cancer risk to make decisions about screening. So you can imagine someone who is trying to decide whether to undergo lung cancer screening, and we can estimate their future risk of developing lung cancer based on their demographics, their smoking history, and we can also envision perhaps doing a biomarker test at this point. Next, please. And we know that the higher this person's future lung cancer risk is, the more likely they are to benefit from screening. So we can use this to choose the people at highest risk, ultimately to be screened, and make that decision about whether this screening should be initiated for this person. Next, please. So let's say they do decide to undergo screening, and now we have more information because we have a CT scan, and we can think about updating our estimate of their future lung cancer risk. And again, at this step, we might envision integrating information from a biomarker test. Next, please. And we can make various decisions at this point using all of this information, such as how should we manage any lung nodules that we identified on the scan, or how long should the screening interval be? Next, please. So in the Lung Cancer Cohort Consortium, we are aiming to develop a custom protein biomarker panel for use in this exact setting, determining eligibility for lung cancer screening and optimizing management during screening. This work is funded by a large U19 from the NCI, which is called Integral, the Integrative Analysis of Lung Cancer Risk and Etiology. And the U19 includes 3 projects. The first is focused on genetics, which is led by Chris Amos. Project 2 is the Lung Cancer Cohort Consortium, which is led by us. And Project 3 is focused on translating all these results into screening studies, and this is led by Regine Hung. Although Project 2 and Project 3 are actually working quite closely together on this panel. So in broad strokes, the project has 3 major phases. The first is a full discovery phase where we're doing a proteomic scan of 1,200 proteins. We then narrow down to a targeted discovery phase of 450 proteins, and then ultimately need to select only 21 proteins for a custom panel, which we will then validate in the consortium. Next, please. This schematic is showing you the same thing, but with more details. So again, we have a full discovery phase, a targeted discovery phase, and then a validation and risk prediction phase using the custom panel. The contribution of the cohorts is shown in the blue boxes, but then underneath in the sort of pink boxes is the contribution of the screening studies from Project 3. So you can see that we're using information at each stage from the screening studies as well. As far as the cohorts go, In the full discovery phase, we measured 1,200 proteins in 2 prospective cohorts, and we completed that about a year ago. And then in the targeted discovery phase, we narrowed down to 450 proteins, which we measured in 4 additional prospective cohorts. And we received those data in March. All of this was using a case control design. And now we are finalizing the task of choosing 21 proteins to include in this custom panel, those that associate strongly with lung cancer risk. And in the future, we will validate this custom panel in all of the cohorts in the Cohort Consortium. So this is about 15 cohorts, but we'll actually switch the design in the final phase from a case control to a case cohort to enable us to really evaluate whether, if we were to use this panel to optimize definition of lung cancer screening eligibility, how would that change whether people are selected or not? Next, please. The platform we're using is from Olink Proteomics. These are high throughput multiplexed immunoassays that use a technology that allows quite specific measurements of proteins in a relatively low volume of plasma. So this makes it ideal for the cohort setting. As I mentioned, we started out by measuring all of the panels that are available at Olink, and then narrowed down as we acquired data. Next one. I'm showing you here the study population for the full and the targeted discovery phases combined. We selected lung cancer cases occurring among current and former smokers, and those occurring within 3 years of blood draw. So these are cases with relatively short lead time. And then we matched them individually to controls for the discovery phases. The cohorts participating in the full discovery were EPIC and the Northern Sweden Health and Disease Study. And then in the targeted discovery phase, we had the Melbourne Collaborative Cohort Study, the Singapore Chinese Health Study, the Hunt Cohort from Norway, and the Cancer Prevention Study too. Next one. This volcano plot is showing you the main results from the full and targeted discovery phases. The full discovery is in blue and the targeted discovery in red. The x-axis is the odds ratio for lung cancer risk, and then the y-axis is the p-value. So we were really quite happy with these results because we found that 20 proteins were successfully replicated in the targeted discovery phase, meaning there were 20 proteins that were statistically significant in both phases. And as you can see, most of these proteins are showing positive associations with lung cancer risk, which is what we expected to see. Next one. We also did fairly detailed stratified analyses, especially for the proteins that seem to be interesting. I'm showing you 2 examples of stratified analyses for different proteins here, and these were 2 of the proteins that were very strongly associated. For these proteins, you can see that the associations were quite consistent across the different cohorts, although this was not the case for all proteins. We frequently saw some heterogeneity by lung cancer histology, and one of the interesting patterns for many proteins was by lead time, where we saw that the association became stronger as time approached lung cancer diagnosis. And this is again what we expected to see. Next, please. So it's been really quite an interesting challenge to narrow from 1,200 to 21 proteins for inclusion on this custom panel. And in the process, we have prioritized proteins that are selected by lasso regression in order to not choose 2 proteins that are giving redundant information. But we also like to see, for a protein that's selected by the lasso, that it shows some reasonable association with lung cancer risk when examined on its own. We also looked for consistent associations across the different cohorts, while allowing for proteins that were specific for certain histological types or were particularly associated close to diagnosis. Sometimes we incorporated external evidence, such as from Mendelian randomization analysis. And as I mentioned earlier, we also have information coming from the studies and the screening studies in Project 3, which will allow us to identify proteins that identify between malignant and benign nodules detected on lung cancer screens. Next one. So after we finalize the definition of this custom panel and it is in the technical development is done, we will validate this panel using a case cohort design across the whole lung cancer cohort consortium. So again, we'll be analyzing lung cancer cases diagnosed within 3 years of blood draw. But in this phase, we will select 2-to-1 sub-cohort representatives, which are sampled at baseline. We'll divide the cohorts into a training set and a testing set, such that we use the training set to develop a score using the biomarker panel and then validate that score independently in the testing set. Next, please. This is our study timeline. So we're sitting here in November 2020, and we've been really happy with the progress we've been able to make over the last year since the last time we were together in Rockville. And this is really thanks to the contributions, especially of the cohorts participating in the discovery phases, because there's obviously been a lot of logistics related to sample shipments and sending data sets and so forth. In the future, we hope to finalize the definition of this custom panel, organize shipment of the samples to Olink for all of the validation phase cohorts, and then hopefully begin analysis of the data sometime in the middle of 2021. Next, please. In the process of compiling all of the data for the protein analyses, we have also put together a harmonized database of all of the cohort participants in LC3. So to the extent possible, we've been collecting data not just on the cases and controls, but on the full cohorts. And this will assist us with our biomarker analysis, but obviously this is a database that can be leveraged for many other purposes as well. We have 24 cohorts contributing to this, representing over 2 million participants and over 60,000 lung cancer cases. There are data available on lung cancer incidence and mortality, baseline questionnaire data, as well as biomarkers on the participants that were selected. And the harmonization of this data set is nearly complete. So we hope to make this available for analysis securely and remotely via the IARC Analytical Hub, which is a new platform that allows remote access of data, which was sort of alluded to in the last session. We are still working out the particular legal aspects for investigators that are outside of the Lung Cancer Cohort Consortium. But if you are interested in leveraging this database for your research, we would be quite interested in hearing from you so that we can sort of continue to work through these issues with you in mind. Next, please. Some of the analyses that we have concretely planned in this database are focused on validation of lung cancer risk prediction models in different geographical settings and in different population groups. This is of interest because these risk models have been proposed for use in defining eligibility for lung cancer screening. And we are hoping to bring on a postdoc to participate in this work. So if you have someone who might be interested, please let me know. The posting for this was just uploaded to the IARC website yesterday, I believe. Xiaoyu Xu at Vanderbilt is leading an analysis to develop a risk model for Asians, and we're also working with the group at the NCI to develop risk models for African Americans and Hispanic Americans. Next, please. So I want to end by thanking all of the people listed here. This has really been a massive effort involving contributions from a lot of different people. So many thanks to all of you and the consortium, and I'm happy to take any questions.
Thank you, Hilary. We're going to move on to the next presentation for the session. I would like to introduce Dr. Paul Brennan. He is a genetic epidemiologist and the head of genetics section at the International Agency for Research on Cancer in Lyon. And he is involved in several of the NCI Cohort Consortium projects, including the LC3, and he will be presenting today on the HPV3 Consortium. So Paul?
Thanks, Nonye. Can you hear me clearly?
Yes. Yes.
Okay, so let's get the first slide up. Great, thanks. So I'll be presenting this on behalf of the HPV Cancer Cohort Consortium. Next slide. So this Cohort Consortium started over 10 years ago. It started initially as a collaboration between IARC and the NCI with Amy Kramer, who is still incredibly active in the Cohort Consortium. It includes 9 cohorts, along with DKFZ, the German National Cancer Institute, for HPV analysis. Some of the things that we've discovered along the years have been that HPV16 antibodies, they predict oropharynx cancer up to 10 years before the actual diagnosis, the occurrence of the disease. This was initially found in the EPIC study. It was confirmed in the PLCO study that also included repeat blood samples. One of the really interesting things about this biomarker was its incredibly high specificity that we've shown again and again across multiple cohorts. In these two initial cohorts, both studies confirmed that the specificity of this biomarker was over 99%. And also, we subsequently showed that this biomarker predicted anal cancer prior to diagnosis, although it appeared to be less relevant for other anogenital cancers. Next slide. So the current investigations that I'll report on today within the HPV Cohort Consortium were we wanted to investigate how long before diagnosis do individuals with oropharynx cancer become seropositive. Both the PLCO and EPIC study showed up to 10 years, but we were curious if this was even longer. Secondly, what is the sensitivity of the marker for both oropharynx and anal cancer when compared to gold standard tumor tissue markers? So we needed tumor samples from within the cohorts. And the third question was, what was the absolute risk of oropharynx cancer among individuals with a HPV16 E6 positive result? Okay, next slide. So just the first one of these. So within the, we sent out a call to the Cohort Consortium for cohorts to participate in this analysis. 9 cohorts responded and were able to satisfy some initial criteria. They're listed here. Among these cohorts, we were able to obtain biological samples for 743 oropharynx cancer cases, over 5,800 controls. We were also extremely interested in serial blood samples, and we were able to obtain serial blood samples across some of the cohorts for 111 of these cases. And tumor tissue was also available from 277 cases, most of these from the Janus cohort, where they made an amazing effort to collect these tumor tissues. Blood was collected on average 11 years before diagnosis, but up to 40 years before. So cases occurring between 1979 and 2015. And then when we undid the analysis, did the results, 0.4% of the controls were HPV16 E6 positive. So again, confirming this incredibly high specificity of the marker for those that had not developed a HPV-related disease. And 26% of the cases were also HPV positive, were HPV positive. Next slide. So here is the analysis for some of those cases where we had repeat blood samples. So these are the cases that were positive on their first blood sample and remained positive. None of them went negative afterwards. So these were 30 cases, and you can see that they were all consistently positive during the 25-year clinical follow-up. Next slide. These were the cases that were negative on the first blood sample and transferred to being positive at some point afterwards. So these are incredibly interesting because they give us an indication of how long before the diagnosis of oropharynx cancer were people seroconverting. And the median time for seroconverting was 11 years, and the minimum time for seroconverting was 6 years. And there was one individual that seroconverted 30 years before their subsequent diagnosis of oropharynx cancer. And the direction, as you can see, the direction of these antibody responses is upwards for all cases, apart from one that hovered around the cutoff point. So next slide. These are the results for anal cancer. So again, all 9 participating cohorts provided blood samples for anal cancer. There were a total of 387 anal cancer cases. There were serial blood samples for 95 of these cases, and tumor tissue was collected on 132 cases. Again, most of these from the Janus cohort. And blood was collected on average, again, 11 years before diagnosis and up to 39 years before. 85 of these cases were HPV16 positive overall. So next slide. So these were the 12 cases that were positive on their first blood sample and remained positive throughout. Next slide, please. 24 of the cases seroconverted during the follow-up period, and the median lead time for the seroconversion was 8 years. Next slide. So now we're talking about the sensitivity of how good this serology analysis was for picking up what we think are true positive or true HPV positive cases. So this work is still underway for the tumor tissues collected for oropharynx cancer. Some initial results from DKFZ on this series here that looked at this series of 277 cancers. 72 of them were HPV positive based on the gold standard tumor tissue result. Of these 72, 66 were E6, were serology positive. So this gives us quite strong evidence that the sensitivity for this biomarker is around 90%, it's in the 90% range for oropharynx cancer, and above 99% for their specificity. So again, this is really quite an incredible biomarker for detection of oropharynx cancer. Next slide. For anal cancer, the analysis has progressed further. So 89 of these 127 cases, of these 127 tumors, were HPV16 positive based on the tumor results. A further 11 of them, or 9%, were HPV positive for other HPV types. And 27 of them, or 21%, were HPV16 negative. When we compare that to the actual serology result, we can see that for those who, where the blood sample was collected within 5 years of the actual diagnosis of anal cancer, the sensitivity of the antibody test was around 73%, or 72.7. Where the blood sample was collected more than 5 years from the anal cancer diagnosis, the sensitivity was only 6%. So here we see a different result to what we see for oropharynx cancer. And the sensitivity of this biomarker really seems to be tied to how close the blood sample is collected to the diagnosis. Next slide. So in summary, the specificity of HPV16 E6 antibodies is extremely high. We've shown this in over 5,000 controls. The sensitivity appears to be around 90% for HPV positive oropharynx cancer. It's lower for anal cancer and is largely predictive of anal cancer within the subsequent 5 years. And seroconversion for oropharynx diagnosis appears to be around 12 years prior to the actual diagnosis, but it can be up to 30 years. Next slide. So the last thing I want to report on, and this is some work that's been led by Hilary, is on the absolute risk for oropharynx diagnosis after a serology test. So our question here is if we were to test a population using this test, and if someone turns up positive for HPV16 E6 antibodies, what is their risk, their 5-year risk or their 10-year risk of oropharynx cancer? So next slide. These are some of the inputs that we used to calculate this risk. So the odds ratios that we've calculated based on the relevant risk factors, the age-specific rates for oropharynx cancer using the USA SEER and CDC data. So these were all incorporated to provide and calculated within the PLCO model to predict what the risk was, the 5- and 10-year risk was of oropharynx cancer based on E6 status. Next slide. So these are some of the results. So this is the 10-year risk of oropharynx cancer for a negative HPV16 E6 serology result. And it's around 0.2%. It was a maximum of 0.2% for smokers. And it's a far lower even for never smokers based on age up between 50 and 70. Next slide. So for HPV16 positive, however, it's clearly a different result. And whether you're a smoker or a non-smoker, it doesn't make any difference here because the risk is completely determined by HPV status. And the risk, the 10-year risk can reach up to well over 15% for age based on a baseline age of 60 or 70. Next slide. So even just looking on the 5-year risks, the 5-year risks for men range between 5% and 10% based on your baseline age. And for women, they're lower, based on around 1.6% to 2.8%, but substantial risks. Next slide. So what does this tell us about HPV16 E6 and oropharynx cancer? So the 5-year risks are between 5 and 10% for men, maybe 1 and 3% for women. And if you look across other types of cancer, these risks are generally consistent with rescreening at short intervals for other cancers, or perhaps even diagnostic workup for older men. So one of the questions we've been posing and thinking of how we're going to move forward with the HPV Cohort Consortium is if we were to rescreen at a shorter interval, we need a non-invasive method. We need another way to pick up if an OPC clinical disease is imminent or not. Next slide. So we've been thinking about how to progress with the Cohort Consortium. And some of the questions we've been asking are, can we identify circulating proteins that predict imminent development of HPV-related oropharyngeal or anal cancer among people who are seropositive for HPV16 E6? And we think some of the results that are coming out of some of the proteomic results that are coming out from the Lung Cancer Cohort Consortium that Hilary just presented are incredibly interesting and stimulating for this. And we think that it does suggest that there could possibly be similar proteomic effects for that could help us identify imminent HPV-related cancer. The other aspect we're interested in is that is with respect to circulating free DNA, because there have been very strong case series results that show that circulating HPV DNA can be detected with quite a high sensitivity for oropharyngeal cancer. And so we're very curious to identify whether circulating free tests for HPV DNA can help us identify the imminent progression of a HPV-associated cancer. And finally, we're interested in whether these circulating proteins or circulating HPV DNA could help predict prognosis for these cancers. Next slide. So I'd just like to finish by acknowledging all of my colleagues at IARC who've helped with this analysis and helped with the progression of this cohort over the last, of this cohort consortium over the last 10 years, with colleagues in the Janus cohort who've been incredibly stimulating and who've provided an awful lot of the tumor tissue as well for this work. Amy Kramer and Alan Hildesheim at the NCI, as well as our wonderful colleagues at the German Cancer Research Center, and all of our collaborators from the 9 participating cohorts. And I'd just like to acknowledge as well the funding from the NCI. So thanks very much.
Thank you both for your updates. I haven't seen any questions come in. We have time for maybe a couple. I'll start off by asking Hilary. I noticed you had planned some analyses in the LC3 database to develop or validate risk models in several different racial or ethnic groups. I'm interested if you can comment on the distribution of race and ethnicity in the study population for which your biomarker panels are being developed, and whether it's a representative target screening population.
Yeah, sure. So, 2 things, I guess. We've done our best to include some of the cohorts that have strong representation of minority groups. Obviously, this is incredibly important. So we do have a few cohorts in the consortium, such as the Southern Community Cohort Study, that have strong representation of minority groups. Another thing we can achieve is combining the small amounts of minorities across 24 different cohorts. That actually gets us a long way for some purposes. It's obviously a challenge for all of us because these cohorts are largely non-Hispanic white, and we kind of are trying to cobble together as much data strength for minorities as we can. I guess I would just say that this is sort of a continuing focus, and we're still looking for opportunities, especially for these data only secondary analysis projects, to add to our database some more cohorts with strong minority representation.
Okay, thank you, Hilary. Paul, there's a question for you in the chat here. For oropharyngeal cancer, do you think the male or female difference is biological or some missing confounders? And I kind of want to add on to that, just noticing that given your study populations, you might have a limited number of minorities. To be able to look at race and ethnicity in your consortium, and if there's any plans to sort of expand the consortium to include more diversity in your study.
Yeah, thanks, Nonye. I mean, it seems pretty clear that the the attributable risk of HPV for oropharynx cancer is higher among men than it is among women. And I think that's what we're seeing reflected there in the actual kind of risk evaluations for you know, if you are HPV positive. So it's, you know, I guess there's just a greater burden of well, let's think about this. So if you're HPV positive, I guess there's a greater amount of false positives among the women, which results in a lower risk. I think that would be my initial take on that question. With regards to the Cohort Consortium, thinking of moving this ahead, you know, we are coming up with these hypotheses of how we can refine these risk estimates based on other biological markers. And so we are going to come back to the Cohort Consortium with these proposals. And the initial proposal was submitted about 7 years ago. And I think the Cohort Consortium has changed an awful lot and it's expanded. And so, you know, we really do need to try and incorporate other cohorts that have different population structures, that have different characteristics of minority populations in them that we weren't able to include at the time. So we are going to be coming back to the Cohort Consortium. We're going to try and think of how we can be more inclusive in the next phase of the analysis on this. So yeah.
Thank you. Thank you, Hilary. Thank you, Paul. I see that we are 2 minutes past our time, and I think we stand in the way of, you know, between you and lunch or breakfast or probably bedtime for some of you, depending on where you are. But I do want to thank you all for joining us over the past couple of days. I would like to hand this over to Lynn to wrap us up. And I do want to thank my co-moderator, Jeannette. Thank you for co-moderating with me. And before I do hand it over to Lynn to wrap us up, I do want to say another thank you to all of you. This is my last meeting of the Cohort Consortium, at least organizing it on this end. It's been a great pleasure to work with all of you for the past 14 plus years. And we'll definitely miss all of you and hope that we will, you know, our paths will cross in the future, particularly around data sharing, which has been a hot topic lately. And hopefully I'll be a part of, you know, the team to help find some solutions or strategies for tackling some of these things. And, you know, a big shout out to my partner in crime, Camille Pottinger, who would be the new face, not new, but kind of assuming the role moving forward. And to the rest of my NCI colleagues and Kathy and Joanne and Scott, thank you all. And I wish you all the best in the future. And Lynn, I will pass it on to you to wrap up and introduce our incoming chair as well.
Yes. Thank you, Nonye, and, you know, thank you for all your service. And I just wanted to thank everyone, all the speakers, all the moderators, and the participants for making, I think, this a very successful first virtual Cohort Consortium annual meeting. And I think it went very well. And I think it's because of all the participation. I want to also thank the steering committee this year, and especially the leadership from NCI. They made being the chair just very easy and actually quite enjoyable. And so I thank them for my time as chair. And I'd like to turn it over to the new chair, which is Roger Milne from Australia. There, he can tell us a bit about what he has planned.
Thanks, Lynn. I'll be quick. It's just past 3 a.m. here on Wednesday the 18th, and I started at 5 a.m. yesterday. So that puts me in about the same headspace as I usually am at one of those meetings in Shady Grove at about 4 p.m. after 10 coffees. I'll try and stay coherent though. So just as a bit of background, I'm the head of cancer epidemiology at Cancer Council Victoria, which is an independent charity, kind of like the ACS, but in Australia, which is a small island at the rear end of the world. I'm PI of the Melbourne Collaborative Cohort Study, which is through which we participate in the consortium. I'm excited taking on the role of chair this year. I've been on the committee for 3 years now and have seen firsthand all the volunteer work that goes behind the scenes to keep the consortium going. And it was suggested that I present here briefly at the end what I'm planning to focus on from the strategic plan in 2021. I'm still thinking this through, particularly after the discussion about the GDPR and its influence on the data sharing for projects. But I was leaning towards the goal of research facilitation. So addressing, or at least starting to address, the issue of duplication in efforts to pool data across cohorts. And I guess at a minimum, I think this would include, and this is from the strategic plan, generating an inventory of which project groups have harmonised what data and from what cohorts, and possibly tying that in with the work from the diabetes project that was a bit of a flagship for standardising some of the harmonisation. I guess looking at sharing, returning, or publishing harmonisation code, and exploring how data from different project groups might be combined. I think that's starting to come up, where we have projects groups that have done a lot of work to pull data, might have pulled epi data, but their genetic data is sitting somewhere else with different data use agreements. And the idea of doing a G by E analysis that a lot of people would like to do is quite a challenge. So really, I'll take this back to the steering committee to get their feedback on it. I would like to talk about the GDPR. That was quite a discussion that requires some work. I don't know whether that's best being led by someone in Australia at an independent organisation, but we'll look into that. We'll also continue to work with plans to come up with guidelines principles on the preparation of DUAs or MTAs. And I guess just lastly, if anyone's interested in these issues, please let me know. I promise not to burden you with work unless you ask me to, but it'd be good to hear your ideas and experiences. And I look forward to working with the committee over the year.